Hi, welcome back to My Smart Learning. This is a lesson for Year 12s and 13s, your A-level biology content. It's useful for Year 11s as well if you're interested in, if you're doing separates in biology and you're interested in doing A-level biology. We started last lesson looking at the beginning, the first stage of respiration. We looked at glycolysis last time. We're looking at the next stage today, which is the link reaction and the Krebs cycle. So how do I release energy to survive is our learning objective for this little section. But before we start, we need our virtual high five because you can't buy those in the shop. So your 5 questions. Number 1, what is the first stage of glycolysis? Number 2, what is the second stage of glycolysis? Number 3, what is the third stage of glycolysis? And number 4, what is the fourth stage of glycolysis? And the last question, number 5, where does respiration occur? So press pause, have a go quickly answering those questions. Once you're ready, you can unpause your video and I'll go through the answers now. So if you're ready, what is the first stage of glycolysis? Well, the glucose molecule, if you remember, gets phosphorylated. The second stage is where the glucose molecule gets split into 2 lots of TP. The third stage is where the glucose molecule then from the TP will produce pyruvate. If I'll just show you back again, a little bit quicker. So the first stage was the activation of glucose becomes phosphorylated. The second stage gets split into TP, like we just said. The third stage is where you get oxidation of the triose phosphates. What do I mean by oxidation? They lose 2 hydrogen atoms. And the last stage is you start producing ATP and you end up with pyruvate. And the last question was, where does it occur? It happens in the cytoplasm of the cell, and therefore it's the same for anaerobic respiration and aerobic respiration. What do I need to know today? Today we're looking at what happens during the link reaction. We need to explain what happens through the Krebs cycle and describe the nature of hydrogen carrier molecules and explain the role of the Krebs cycles. So before we can get onto that, we need to know what this phrase means. What are coenzymes? Well, you come across enzymes before. Enzymes are proteins that bring about chemical reactions in biological systems, biological living things. Now, for some enzymes to function, they need chemicals to bind to them before they become activated. So that's what these molecules are called. They're called coenzymes. They allow enzymes to function. Photosynthesis and respiration are both biochemical pathways requiring enzymes for their reactions to occur. Now, these enzymes don't usually just function on their own. They need other chemicals to bind to them so that they can function efficiently. And that's what these coenzymes are. So they are not enzymes in their own right. They are substances that bind to enzymes so they can function. The most important one for respiration is NAD. NAD is a coenzyme and it works with dehydrogenase to remove hydrogen molecules. So like we talked about oxidation and reduction. Now reduction is the gain of hydrogen, which means if you're oxidizing something, you're removing the hydrogen. So if oxidation is the removal of hydrogen, reduction is the gain of hydrogen, well, dehydrogenase is an enzyme that removes the hydrogen. So if one thing's being reduced, that means something else is being oxidized. Okay, so that's what dehydrogenase does, and NAD happens in glycolysis, and you'll see it happens in the Krebs cycle, and you need them in the electron transport chain. So it happens throughout and also in the link reaction. NAD is important throughout the entire respiration phases. FAD is another one of these coenzymes, and you'll see it mentioned in the Krebs cycle in a minute. And in photosynthesis, when we talked about NADP, that was a coenzyme that becomes reduced, and that's why it becomes NADPH. Again, photosynthesis can't happen without this coenzyme. So, what is the link reaction? The link reaction is where, remember, the product from glycolysis is pyruvate. So what happens to this pyruvate? This pyruvate then is actively transported into the cristae of the mitochondria. So if you remember the structure of mitochondria, they look like little beans, little sort of bacteria-shaped beans, but on the inside they've got double membrane, they've got these little folds inside. Those little folds are called cristae, and those cristae have little enzymes all bound to them, attached to them. The matrix is the liquid cytoplasmy stuff that's in the middle of the mitochondria. So pyruvate is actively transported into the cristae of the mitochondria. Then what happens to the pyruvate then, once it's basically in the mitochondria, because remember glycolysis is in the cytoplasm, the rest of photosynthesis is happening inside the mitochondria for aerobic respiration. This pyruvate is oxidized. What does that mean? It loses hydrogen. So if the pyruvate loses hydrogen, this hydrogen needs to go somewhere. Well, it's going to join onto this NAD. So the NAD becomes reduced, and therefore that's why it becomes reduced NAD, because the hydrogen has jumped from that one onto that one. So it loses 2 hydrogens and jumps onto that one, because the pyruvate is being oxidized. Whenever something's being oxidized, something else is being reduced. The NAD here is being reduced. The pyruvate also loses a carbon dioxide molecule. So whatever's left from this is now known as acetate. So this becomes acetate. But the acetate will combine with this coenzyme A. So it's another coenzyme. So this coenzyme A, once the acetate joins the coenzyme A, you get this acetyl coenzyme A, or acetyl coenzyme A, whichever way you want to pronounce it. So our products then are acetyl coenzyme A, reduced NAD, plus carbon dioxide. It's our first loss of a carbon dioxide. Okay, so now if you think about it, there's going to be 2 link reactions, because one pyruvate, but how many pyruvates do I get from my glycolysis? Because remember, glucose gets split into 2 lots of TPs, and you get 2 lines going down. So therefore you've got one pyruvate, another pyruvate. So if you've got one pyruvate, you get 1 carbon dioxide. Then the other pyruvate joins on here. You've got 2 carbon dioxides being made. Now, one of the biggest misconceptions of respiration is that you automatically assume this, and this is probably because of GCSE. Glucose plus oxygen gives you carbon dioxide plus water. You think plus oxygen. That oxygen must be joining onto the carbon in the carbon atoms in the glucose molecule to make carbon dioxide on the other side. That's not what happens. That oxygen that you're breathing in for respiration is nothing to do with that carbon dioxide there. The oxygen you'll see in the next lesson on electron transport chain is a way of mopping up the electrons at the end and the hydrogens at the end to make water, so that you don't die from acidosis. The oxygen is a way of cleaning your system, is to mop up all the poisons so that you don't die. That's why if you get strangled, the lack of asphyxiation, lack of oxygen, you'll die from acidosis. It's not the oxygen joining onto carbon to make carbon dioxide. That's not what's happening. So carbon dioxide actually comes from the glucose molecule, because remember, glucose is C6H12O6. There's carbon in there and oxygen in there. It's a carbohydrate. Carbo meaning carbon, hydrate meaning the hydrogen, ate meaning the oxygen. You lose a carbon and an oxygen from the glucose, it breaks away. So from each pyruvate you lose 1 carbon dioxide, but because you've got 2 pyruvates, you're going to lose 2 carbon dioxides. So I'm going to have to lose 4 more from somewhere, because remember, you get 6 CO2s. So that is the link reaction then. Your pyruvate loses hydrogen. The hydrogen will join onto NAD, so it becomes NADH, or reduced NAD. You will lose a carbon dioxide from there, and what will happen is whatever's left over is called acetate. That will join onto the coenzyme A to make this product here called acetyl coenzyme A. And that is the important product, because if you remember our flow diagram from our previous lesson, that is the important product from the link reaction. So, to summarize then, there's the pyruvate. It's a 3-carbon molecule. The reduced NAD, the NAD becomes reduced because a hydrogen from there, or 2 hydrogens from there, will join onto that to make reduced NAD. You lose one of the carbon atoms there because that becomes carbon dioxide. You're left with a 2-carbon compound called acetate. That acetate then will join onto coenzyme A and therefore will become acetyl coenzyme A or acetyl coenzyme A. That is the important thing that jumps onto our next stage known as the Krebs cycle. Krebs cycle. So what happens during the Krebs cycle? Well, here's my 2-carbon compound. It's going to jump onto this conveyor belt. It's a bit like in the airport where it goes round and round and round. But if you notice, there is a 4-carbon molecule that exists on this Krebs cycle, and it keeps going round and round and round and round. Now for the AQA syllabus, you don't need to know the name of this actual 4-carbon compound. You can just call it a 4-carbon compound. However, if you do other exam boards, for instance Excel or OCR, I think for those examples you need to know the actual names of these compounds that are going around here. But for the AQA, 4-carbon compound is fine. So the acetyl coenzyme, then there's 2, will join onto this 4 there. You'll end up with a 6-carbon molecule. So this 6-carbon molecule then will now, will lose more hydrogen atoms. They will join onto this NAD there and this FAD there. So you'll end up with reduced NAD and reduced FAD. They're very important because they're the guys that we need for the next stage, which is the electron transport chain. So those 2 is what we need for the next stage, electron transport chain, where we get lots and lots of ATP. But this 6-carbon compound breaks away. You break off one of those carbons, you break off the second carbon. They end up becoming carbon dioxide. Not because it's joined with the oxygen you breathe in. Remember, sugar, glucose, C6H12O6 has got carbon, it's a carbohydrate, it's got carbon, it's got oxygen. The carbon and the oxygen break off. So you lose 1 carbon dioxide, 2 carbon dioxides. So you also get an ATP being generated. Again, this is known as substrate level phosphorylation. Substrate level, because this is happening at a substrate level. There's no redox going on here with this particular ATP. Okay, so it's the substrate level phosphorylation, and what you eventually get then is it will get recycled and go all the way back because you've lost 2 carbons to make carbon dioxide, and therefore the 4-carbon molecule will start back from the beginning again. Acetyl coenzyme A will join on, and then the whole thing goes round and round and round. So where does my 6 carbon dioxide come from respiration? Well, I've lost 1 there, I've lost 1 there, I've lost 1 there. That's only 3. So how come I get 6 CO2s from my devil's equation? Because you don't get 1 pyruvate, remember, you get 2 pyruvates from glycolysis. So all of this happens twice for 1 glucose molecule. So 1 pyruvate, 1, 2, 3, second pyruvate, 1, 2, 3. Therefore you got your 6 carbon dioxide without no mention of oxygen at all from you breathing in from aerobic respiration. That happens right at the end, and that's at the end of electron transport chain, oxidative phosphorylation, which is going to be your next lesson. So in summary then, in today's lesson we looked at the link reaction and we looked at the Krebs cycle. This is happening in the matrix then. This bit's happening in the matrix of your mitochondria. Okay, you got the first bit in the cristae, then the mitochondria matrix will have your Krebs cycle. And what we're going to look at next time is, after we finish the Krebs cycle, is the electron transport chain, because those guys, the reduced NAD and the reduced FAD go off, and this one here, the reduced NAD there, remember, and you've also got the reduced NAD from the glycolysis, because we get 2 from there. They all go off to the electron transport chain, is where the big factory is of making the ATP. Because if you think, we only got 2 from net effect from glycolysis, and we've only got 1 for every turn of this cycle, but we get 2 pyruvates, so we've only got 2. So far we've only got 4 ATPs. So it's again not that great. it's the electron transport chain that's really important for generating lots and lots of energy from ATP. So why is the Krebs cycle so important? Because it breaks down what we call macromolecules, so large molecules into lots smaller and smaller molecules. Okay, you get lots of hydrogen atoms then, and those are carried by NAD to the electron transport chain, not etc., not ETC as in etc. Electron transport chain. So the NADs become reduced by those hydrogen atoms. That's the important bit. That's where the energy is being made. It regenerates the 4 carbon molecules with acetyl coenzyme A. If you didn't constantly get this Krebs cycle, this 4 carbon molecule will eventually build up and the whole thing will come to a grinding halt. And like we was talking before, it's a source of immediate compounds in the cytoplasm to make things like fatty acids, amino acids, and chlorophyll. So the Krebs cycle is really important for these kind of compounds that you get made, these coenzymes, to help go and make other compounds in the cytoplasm. Okay, but remember this is happening inside the mitochondria. So some questions for you guys to have a go at. It's on the PowerPoint. I will share it on Show My Homework. Make sure you're doing your exam style questions, the summary questions, like I've been putting them on the slideshows now. So they'll be on Show My Homework. Miss Martin's going through chapter 9 and 10, finishing off those. I'll finish off the respiration chapter this week and next week, and then we'll probably start pausing for a little bit just so you can catch up with all your learning. Hopefully you learned something from my smart learning today, and if you got all your 5 questions right from your starter activity, here's your virtual high five. See you next time.